Hello students, hope so you are doing well at your home and safe at your home. Today there is the first lecture of pharmaceutical microbiology. So in today's lecture we are going to study about the introduction to pharmaceutical microbiology. The contents of my today's presentation are syllabus, then introduction, history, branches, scope and importance of pharmaceutical microbiology. Firstly, we will see the syllabus of pharmaceutical microbiology. Unit 1, the syllabus of pharmaceutical microbiology is generally divided into two categories, that is general microbiology and pharmaceutical, or we can say industrial microbiology. The general microbiology covers the study of various microorganisms like bacteria, fungi, viruses, and the other part, that is industrial microbiology, mainly carries the various pharmaceutical operations, that is sterilization, fermentation, then microbiological assays, disinfection and all. So unit 1 contains the introduction, history of microbiology, its branches, scope and importance, the prokaryotes and eukaryotes, then the ultrastructure of bacteria, morphological classification of bacteria, then what are the different nutritional requirements for the growth of bacteria, raw materials used for the culture media, physical parameters for growth curve, isolation and preservation methods for pure cultures, cultivation of anaerobes, quantitative measurement of bacterial growth, then in detail study about different types of microscopy and definition and examples of probiotics and prebiotics. Unit 2, the identification of bacteria using various staining techniques or methods, that is simple staining, gram staining and acid fast staining, and different biochemical tests. Then definition of D and Z value, it is related with sterilization, then various methods of sterilization, then evaluation of efficiency of sterilization methods, then equipments employed in large scale sterilization, and sterility indicators. Unit 3, morphology, classification, reproduction and cultivation of fungi and viruses, then classification and mode of action of disinfectants, factors affecting disinfection, antiseptics and their evaluation for bacteriostatic and bactericidal actions, sterility testing of products according to IPBP and USP. Then unit 4, designing of aseptic area, laminar flow equipments, study of different sources of contamination in aseptic area and methods of prevention. Then clean area classification, principles and methods of different microbiological assay, methods of standardization of antibiotics, vitamins and amino acids, and assessment of new antibiotic. Unit 5, types of spoilage, then factors affecting the microbial spoilage of pharmaceutical products, sources and types of microbial contaminants, assessment of microbial contamination and spoilage, preservation of pharmaceutical products using antimicrobial agents, then evaluation of microbial stability of formulations. Then growth of animal cells in culture, general procedure for cell culture, primary established and transformed cell cultures, application of cell cultures in pharmaceutical industry and research. So this is the syllabus. Now we will see in detail means the introduction of pharmaceutical microbiology. The word microbiology was derived from the Greek, which consists of micro means very small, which cannot be observed with our naked eyes, and biology means life. So in short, the microbiology is the study of microorganisms. Microorganisms means those organisms which are of microscopic size. For their observation, the microscope is mainly required. So, pharmaceutical microbiology, or we can say only the microbiology, is the branch of science that is concerned primarily with the study of microorganisms and their effects on other living organisms. For example, bacteria, protozoa, algae, fungus, and viruses. So, these are the groups of microorganisms which we are going to study in detail. So, that is the general microbiology. The microbiology may be pure or applied. Pure microbiology includes bacteriology, mycology, protozoology, phycology, parasitology, and many more. While the applied microbiology includes medical microbiology, pharmaceutical microbiology, industrial microbiology, and many more. When microbiological concepts, processes, and techniques are applied to pharmaceutical operations, the subject is then called as pharmaceutical microbiology. As we are dealing with the study of pharmacy, so we have to study in detail about pharmaceutical microbiology. So all those microbiological concepts are mainly applied to the pharmaceutical operations, and that's why the subject is called as pharmaceutical microbiology. Firstly, we will see about the history of microbiology. There are various scientists who mainly contributed into the development as well as various discoveries of the microbiology. The first scientist is Antony van Leeuwenhoek, who was the greatest microscopist. Microscopist means who mainly work for the subject of microscopy, means it is the science of microscopes. So microscope is the device, or we can say it is an equipment which is mainly used to observe various minute things which are of microscopic size. So as we know that the microorganisms are of microscopic size, so microscope is mainly required. So this microscope was actually discovered by Antony van Leeuwenhoek. So he was the first person to actually see living microorganisms. He was the first person to observe human RBCs. He described and sketched different shapes of bacteria. He mainly assembled 250 simple microscope. The glass grinding and preparation of lenses was his hobby, and this hobby led him to assemble different 250 simple microscopes. So the contribution of Antony van Leeuwenhoek was very great, and from his discovery, then the world of microscopy likely to begin. Then the next scientist, Louis Pasteur, who was the French organic chemist. Now he is considered as father of microbiology because number of discoveries were carried out related with microbiology by him, and that's why he is mainly considered as he is mainly considered as father of microbiology. So various discoveries were carried out by him, that is fermentation process. The fermentation process is the process which is mainly used for production of various useful compounds by the use of microorganisms. Then he also found out that microorganisms cause the spoilage of various foods as well as various other things. He demonstrated the gun cotton experiment, means in the experiment mainly consists of he demonstrated the gun cotton experiment, and from that he concluded that the air contains microorganisms. So for that he passed a large quantity of air through a tube, that contained a plug of gun cotton to serve as a filter. The gun cotton was then removed and dissolved in a mixture of alcohol and ether, and the sediment was examined microscopically. He found that this sediment contains not only the organic matter but also a large number of small microorganisms. Then he he also concluded that at different places there are a different concentration of microorganisms. Means the number of microorganisms at particular place is not constant. Then he designated life as aerobic, anaerobic, and facultative anaerobic. Aerobic means the life which is possible in presence of oxygen. For example, aerobic bacteria. The aerobic bacteria require the oxygen for their growth as well as survival. Anaerobic bacteria means which do not require oxygen for their growth as well as survival. If oxygen is present, then that oxygen is toxic to such kind of bacteria. And facultative anaerobic bacteria or microorganisms means these can either grow in presence of oxygen or in absence of oxygen. So all these concepts were designated by him. And he opened the field of sterilization. The sterilization is very important in pharmaceutical point of view. So sterilization is the process by which the various things are made free from microorganisms. It is a process to kill the microorganisms. So this concept of sterilization was also discovered by the Louis Pasteur. There are various methods of sterilization. For example, the dry heat sterilization, moist heat sterilization. These methods of sterilization were also discovered by him. The different equipments, for example, autoclave as well as use of hot air oven for sterilization was also discovered by the Louis Pasteur. Then he also developed the pasteurization process. The process of pasteurization is also, we can say it is a process of preservation, means it is mainly used for pasteurization of milk. He also discovered about the virulence power. Virulence power means it is the ability of organisms to have immunity against various diseases which are mainly caused by microorganisms. He also developed the vaccine for rabies as well as anthrax. Okay, so due to his greatest discoveries, he is mainly considered as father of microbiology. Next scientist is Robert Koch, who was the German physician who is considered as father of medical microbiology because he mainly carried out various researches or various discoveries related with medicine microbiology. He was the first to demonstrate the role of bacteria in causing diseases. Means he concluded that some of the diseases which are infectious are mainly caused by bacteria. He was the first to isolate Bacillus anthracis. Bacillus anthracis is the name of a bacteria that mainly causes the anthrax. He developed staining methods of microbes. Staining methods means application of dye or application of stale to microbial cells, and after that we can observe them under the microscope because microorganisms are transparent, means cells are transparent and they cannot be observed as it is under the microscope. For that purpose we have to apply the stain, and after application of this stain we can observe under the microscope. So this technique was developed by him, that is Robert Koch. He developed or discovered the Mycobacterium tuberculosis and Vibrio cholera. These are also two bacterial species. As the name suggests, Mycobacterium tuberculosis means which causes the tuberculosis, while Vibrio cholera means which causes the cholera. He also awarded a Nobel Prize in 1905 for his greatest discovery regarding tuberculosis as well as cholera. He developed the relationship between microorganisms and diseases. Next scientist is Paul Ehrlich, who is the father of chemotherapy, who mainly discovered the concept of chemotherapy. Chemotherapy means the use of the various chemotherapeutic agents for various purposes, for curing various kind of infections, and that was mainly discovered by Paul Ehrlich. He actually classified various stains as acidic stain, basic stain. Then he developed the staining methods for RBCs. Then he developed Zell-Nielsen staining. He was awarded with Nobel Prize for immunity, study of immunity. Then next scientist is Alexander Fleming. The first antibiotic penicillin was discovered by him. Then Richard Petrie, who discovered the Petri plate. You can see into the figure. Those images are the Petri plates. These Petri plates are mainly used for the growth of microorganisms, and it was discovered by the Richard Petrie, and that's why the plate is mainly called as Petri plate. So all means these are the 4 to 5 or 6 scientists who mainly contributed into the development of the field of microbiology. There are also many more scientists, but these were the main. Now, what are the different branches of microbiology? First, medical microbiology means it is the study of causative agents of infectious diseases. Then pharmaceutical microbiology, it is the study of microorganisms that are related to the production of antibiotics, enzymes, vitamins, vaccines and other pharmaceutical products, and that cause pharmaceutical contamination as well as spoilage. So all these concepts we are going to study into our syllabus, that is pharmaceutical microbiology. Then industrial microbiology, which is the study of industrially useful microorganisms in production of alcoholic beverages, vitamins, amino acids and antibiotics. Then microbial biotechnology, the manipulation of microorganisms at the genetic and molecular level to generate useful products. Means the field of biotechnology is mainly depends upon the microorganisms, and these microorganisms are mainly genetically converted, or they are converted at their molecular level to produce various useful products. For example, various proteins. For example, insulin is a kind of protein that is mainly used for diabetes mellitus. That is also discovered, that is also produced by using the microbial biotechnology. Then food microbiology, which is the study of microorganisms which cause food spoilage and foodborne illness using microorganisms to produce foods, for example by fermentation. Then soil microbiology, which is the study of those microorganisms that are found in soil. The microorganisms which are found in soil are also important because these some of the bacterial species play an important role in fertility of soil. For example, nitrogen fixation is mainly done by some of the bacterial species, and that's why it is mainly important to study, because soil contains number of microbial species and they can be isolated, they can be cultivated, and that is mainly done in the branch of soil microbiology. Then agricultural microbiology, which is the study of agriculturally relevant microorganisms, means there are some of the microbial species which are important for agricultural field. Okay. Then aquatic microbiology, as the word suggests, aquatic, which are means the study of those microorganisms that are found in water. Yes, the microorganisms are also present into water. Water is the rich source of bacteria as well as various microorganisms. So those bacteria or microorganisms can be isolated from water also. That is aquatic microbiology. Then air microbiology. It is the study of airborne microorganisms. Yes, the Louis Pasteur demonstrated that the air contains microorganisms, but which are invisible to our eyes. That air contains number of microorganisms, mainly the bacteria. So their study is mainly included into the subject of air microbiology. Then environmental microbiology, which is the study of function and diversity of microbes in their natural environments. Now, what is the scope and importance of pharmaceutical microbiology that we will see? First, the production of antibiotics. Before understanding the scope and importance, firstly we will see what are the different beneficial roles of microorganisms as well as harmful role of microorganisms. Yes, these microorganisms play the beneficial role, means some of the microbial species are beneficial to human beings, and second, some of the microorganisms are harmful to the human beings. Firstly, we will see how microorganisms are beneficial for human beings. We already discussed that these microorganisms are mainly used for production of various useful compounds. For example, antibiotics, vitamins, alcoholic beverages, then enzymes, various organic acids. These microorganisms are mainly used for production of these useful compounds by the process of fermentation. Okay, so these are the beneficial role. On to the other hand, these microorganisms play harmful role. For example, some of the microbial species cause the diseases to the human beings. Means these microorganisms are mainly called as pathogenic to the human beings, which cause the infectious diseases. And that's why, due to this beneficial role as well as harmful role, these microorganisms are mainly significant for studying into the subject of pharmaceutical microbiology. Okay, and that is mainly there into the scope and importance of pharmaceutical microbiology. And that first one is production of antibiotics. So today's 2/3 of antibiotics are mainly produced from microorganisms. The antibiotics are penicillin, cyclosporins, and many more. Many antibiotics are isolated from natural microorganisms by the process of fermentation. For example, penicillin is the antibiotic which is mainly produced by using Penicillium species. Penicillium is the name of bacterial species and penicillin is the name of antibiotic. Then streptomycin from Streptomyces griseus. Then tetracycline from Streptomyces aureofaciens. Then chloramphenicol from Streptomyces venezuelae. So the antibiotics are mainly produced by the use of these microorganisms, mainly bacteria as well as fungi. Second, production of enzymes, vaccines, and other pharmaceutical products. Many microbial cells convert raw materials or substrates into valuable organic compounds. For example, sugar is mainly converted into the industrial alcohol by the process of fermentation, and fermentation works in presence of microorganisms. These microorganisms secrete various enzymes, and these enzymes are actually responsible to convert the raw material into the useful compound. So microbial cells produce these enzymes, and these are mainly important for the success of pharmaceutical fermentation process. Then diagnosis of diseases and treatment. Different tests are used to detect infectious microorganisms. For example, ELISA is the test which is mainly used for diagnosis of AIDS. Then Vidal test. So number of tests are mainly there which work in the presence of microorganisms. Then fourth, treatment of industrial waste material. Most industrial process produce waste materials, means water, salt, organic matter, and spent media, and this material may be toxic to human beings. So many microbial species are species are used for decomposition of such waste materials and organic components. Then fifth, that is plant growth promotion. Yes, many microbial cells are mainly present into the soil, and they play an important role in soil fertility. Then herbicidal resistance, insect resistance, change in protein or oil content, and enhanced quality of plant products. Herbicidal resistance means resistance for herbicidal action. Insect resistance means resistance of insects. So that role is mainly played by these microorganisms. Then sixth, that is sterile product preparation. Pharmaceutical microbiology plays a major role in preparation of sterile products. Sterile products means those products which are free from microorganisms. For example, various injectable preparations, IV injections, surgical dressings and all, those should be free from microorganisms, and those products are mainly called as sterile products. So that deals with sterile rooms, aseptic techniques, detection of microbes by sampling and sterility testing of different sterile preparations. Then sterilization. It is the process which mainly kills the microorganisms, which eliminates or removes the microorganisms from all forms of life and other biological agents. That is mainly called as sterilization. Moist sterilization, dried sterilization, membrane filtration, gaseous sterilization, chemical sterilization are the methods used for killing microorganisms. Then identification of microorganisms. One of the vital functions of pharmaceutical microbiology is identification of microorganisms found in products and the manufacturing environment. The microorganisms are isolated and identified by morphological, biochemical, cultural, microscopic characteristics and genetic studies. Yes, the identification of microorganisms is one of the important functions of any microbiological laboratory. So these microorganisms are mainly identified by various methods, that is by identifying their morphological characteristics, or by performing various biochemical tests, or by observing various cultural and microscopic characteristics, and that is mainly used for identification of various microorganisms. For example, identification of microorganisms can be done by staining, so that is mainly used for identification. Then testing of pharmaceuticals. Various raw materials and finished products are mainly tested for their detection of various bacterial species which are mainly harmful to the human beings. If these finished pharmaceutical products contains those harmful microorganisms or bacterial species, and if they are administered to the patient, then that such kind of products will be harmful to the patient. Okay, so that's why these products should be free from those bacterial species which are mainly harmful to the human beings. For example, Escherichia coli, Salmonella typhi, Pseudomonas aeruginosa, Staphylococcus aureus, their presence must be detected into these raw materials as well as finished products. Then microbiological assay of antibiotics. Yes, the assay of antibiotics cannot be performed by using titration as well as various physical methods or various chromatographic techniques. The antibiotics can only be assayed by using microbiological method. The antibiotic bioassay provides a potency for the overall biological activity of antibiotic preparation, and there are various methods to perform these microbiological assay. Then evaluation of disinfectant. Effective cleaning and disinfection of manufacturing facilities are important to the achievement and maintenance of high quality standards required for medicine and medical devices. Selection of cleaning agents and disinfectants will be influenced by the number of raw materials, number and type of microorganisms, then maximum residual levels, acceptance on completion. So there are various tests which are mainly used for evaluation of this disinfectant. So disinfectant is the chemical agent that is mainly used to prevent the infection. For example, alcohol, phenol, all those are disinfectants, and these disinfectants are mainly playing very important role into today's pandemic COVID-19 situation. So these disinfectants as well as antiseptics should be evaluated for their potency as well as efficacy. Then antimicrobial preservative efficacy testing. Preservative are those chemicals or materials which are mainly added into the pharmaceutical preparation for preservation of that particular product, and their efficacy should also be tested. Then endotoxin testing. Endotoxin, bacterial endotoxin or pyrogens have a number of physiological effects following intravenous injections, such as fever, activation of cytokine system, endothelial cell damage, and many more. And that's why those endotoxins must not be present into those sterile preparations. Those must be eliminated from those sterile preparations. Then last, test for support of sterility assurance system. Sterility testing includes many of the basic principles applicable in aseptic area and microbiology laboratory that deal with the testing in support of sterility assurance system. The aseptic area means the area which is free from microorganisms. That area should be implemented during for carrying out microbiological assays as well as various pharmaceutical operations. So, aseptic techniques must be followed during such kind of activities. Okay, so all these operations or testing of pharmaceutical are mainly important one in pharmaceutical point of view. So, this is all about introduction to pharmaceutical microbiology. Thank you.